Hi, it's a pleasure to be here to present on Myra Vision. I'm thrilled to tell you a little bit about our system we call the Calibri system. Before I do that, I have to give a big shout out to our creative and dedicated team at Myra that's really worked the last few years to bring this innovation to life. So let me start by giving you a quick company overview. So Myra was founded by ShifaMed, which is a medical device innovation hub based in Campbell, California. We've had 6 successive exits to strategic acquirers, and in that time, we've developed a really new and unique innovation process that we're now applying to the ophthalmology field. And Myra Vision is one of those companies that we're working on. We're focused on glaucoma therapy. We were founded in 2019. We just raised our Series B earlier this year of $25 million. We currently have 25 employees that are housed in Campbell, California. You've heard a lot about glaucoma and glaucoma therapy. It still remains today that IOP is the really only modifiable risk factor to reduce or treat glaucoma. There's lots of different outflow pathways that you've heard talked about previously. The subconjunctival outflow pathway is really the main area that has had the most IOP reduction and treated and had good therapeutic outcomes for those patients. Yet there's still an opportunity to improve. If you look on the left side here in the tube versus trab study, you can see that many patients didn't achieve the outcomes that they would have liked. On the right panel, you see that a lot of that is just due to inadequate IOP reduction. We founded Myra Vision to address these shortcomings. Our system is called the Calibri system, and our focus is really to bring titratable glaucoma therapy to glaucoma treatment for physicians. That allows them to personalize the IOP for each patient. They can also avoid some of the negative outcomes that occur with other therapies, and we're doing this with existing procedures and existing materials that are out there today and being used in glaucoma therapy. How do we do that? Well, we developed an aqueous shunt. Aqueous shunts have been well known. Our aqueous shunt is a silicone-based product with a nitinol valve assembly in the inflow end, as you can see on the left there. It's 10 millimeters long, 1.6 millimeters wide, and about 0.3 millimeters thick. The device is implanted using an ab externo method that's very well known. We open up the conjunctiva, make an incision into the anterior chamber, and then suture the device down. We do use mitomycin, which is very standard in ab externo delivery. The device sits very parallel to the iris in the anterior chamber and conforms to the sclera on the outflow end of it. On the right here, you can see an OCT image of the device where it's sitting very parallel to the iris. We designed the device to really conform to the globe. On the left side here, you can see that the device is much softer than the scleral tissue. And indeed, when you take the device out of the eye, you can see that it retains the shape of the globe after removal. Once the device is implanted, we can then adjust the flow rate using a standard green laser. Typically, we're using 100 millisecond duration, 200 micron spot size, and 300 milliwatt power range. It's a transscleral actuation there. Once you're ready to adjust the flow, you bring the patient to the slit lamp. If you can please play the video. Okay, so when we first implant that device, you can see that the center channel is open, so you get flow immediately upon implant. If you would like to adjust the flow, you can use light energy from the laser to use the shape memory characteristics of nitinol to open the inlet to the lower channel. That allows flow through the lower channel so you can have more outflow through the device. If at any time you would like to close the flow off, you can just hit the other side and it'll close the channel. And the same thing with the upper channel. You can open or close that at will just with a simple slit lamp visit using a standard green laser that most people are comfortable with. We've taken the technology to the preclinical setting. You can see on the left panel here, this is a picture of a device implanted in a rabbit eye. On the right side here, you can see that we're actuating the valves in vivo, and we're getting opening and closing of those valves. And I'll show you in a second here that we actually get flow going through the channels, the valve control channels. And you can see on the left side here is the baseline flow. On the right side is a maximum flow. We've taken the technology also to chronic animals and have demonstrated good biological response. It's all materials that have been used in the eye previously, so we expected to see this, but we're indeed seeing very little reaction to the device as it's in the eye chronically. With all this data, we're now starting our clinical trial. It's a 30-patient clinical trial based in 3 sites. We have 12 months as a primary endpoint where we're looking at procedural success, safety, and efficacy. We're looking forward to continue to advance our titratable glaucoma therapy, and we'll report back as we get more data in the near future. Thank you very much.